Hi, and welcome to this video run-through of the 2017 A-Level Biological Processes paper from OCR. Now this paper is pretty long. This is the first paper we've done for the A-Level length paper, which is 2 hours 15 minutes, as opposed to the AS. So I'm going to try and keep it as brief as possible, but I suggest that you probably watch this video in 2 segments or even 3 segments, maybe 3 40-minute segments. I'll try and keep it as quick as I can. So first thing I'm going to do is I'm going to show you the mark scheme to the multiple choice section. Okay, so I want you to just mark the multiple choice section that you've done, and then I'm going to go through these questions, but you can only just watch, you only need to watch the ones if you're not sure where you went wrong, or the ones that where you went wrong to make sure you understand why. Okay, so pause the video there. And now I'm going to run through these multiple choice questions. Okay, first one. Which of the options A to D correctly describes how an endotherm, remember that's like a human, would respond to an increase in temperature? So this is how would you lose sort of heat energy? Yes, this is correct. So A is correct. No, this is like hair standing up, and that would, if it's cold, rapid, that's shivering. No, sweat glands, less sweat, that's no. So that's simply A. Which of the following correctly summarize photosynthesis? Well, let's look at each part independently. Okay, well, this is correct. H2 goes in and oxygen comes out of the light. Oh, look at this. They've put it the wrong way round. That's really horrible. Okay, so that's horrible. They put it the wrong way round. So you would have thought the light dependent would be on the left, but they've done it the other way around. So that's how they tried to trick you. So this is wrong. Light dependent. Oh, look, they're all in different. Okay, light dependent reaction. H2O in. Reduced NADP out. That is actually correct. That implies it's a waste product, so that's wrong as well. NAD is NADP for photosynthesis, not NAD. Let's try this one. Light dependent. Okay, this is light dependent on the left. H2O going in, O2O coming out. Yes. Reduced NADP. That's correct. That's correct. Light independent reaction. CO2 goes in and hexose comes out. So C is correct. Great. D. Well, that's on the left, so yeah, that's not right. Yeah, okay. So the answer is C. That's a tricky one. Attention to detail is the key. 3. A student counted stomata on using a light microscope. This shows the stomata that are visible. The magnification is 60 times 60. So we've got to count them. Okay, so we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, and 17 stomata. And it's times 60. Now you're going to have to measure this. Measure this length here in millimeters, which I'm not sure because I haven't got a ruler or the ability to use a ruler. And you measure this length in millimeters. But then you've got to divide it by 60 because it's magnification. Then you divide this by 60, and that's magnification. Then you work out the area. So it's 17 stomata in that area. And that will give you the density, which, looking at the mark scheme, is A. Which of the following A to D occurs in the nucleus of a cell? Synthesis of enzymes, no, that's at the ribosome, so that's not in the nucleus. Synthesis of RNA, yes, that's true. Modification of polypeptides, no, that's generally in the endoplasmic reticulum or the Golgi. And synthesis of carbohydrates, again, in the cytoplasm. So the only answer is B. 5. During cell division, the chromosome number in the cell changes. Different types of cell division. Which of the following A to D correctly describes the stages of mitosis and meiosis in human cells? Okay, well, let's have a look. Okay, so 2 to 4 to 2 to N. This is meiosis. 4, this is nothing. That's just false. Through N to 2N to 4N, this is nothing. 2N to 4N to 2N, this is mitosis. Okay, so that's what we work with. 1 is mitosis, 2 is no. So we're basically looking for a 1 and a 4. 4 is Mitosis, 1 is meiosis. Yeah, that's correct, D. Okay, so here we are, question 6. Patients with kidney failure can be treated in different ways. Which of the following statements describes a feature of peritoneal dialysis? That's the one where the fluid is kind of put into the abdominal cavity. So urea and mineral ions pass into the tissue fluid. This is kind of a bit weird the way it's worded, tissue fluid. I would have said dialysis fluid, but I think if they're talking about the fluid, the dialysis fluid, then that's a yes. Blood is passed over an artificial membrane to remove toxins. No, it's not. It's a biological membrane, the peritoneal cavity membrane, I think it's called. And the patient doesn't have to receive immunosuppressant because it's not a transplant. So just 1 is true, I guess. So it's got to be D. 7. Bony fish absorb oxygen from water using gills. Water is passed through the buccal cavity and over the gill lamellae. The oxygen saturation of the blood and water changes as the water passes over the gills. Yes, that's true. All right, which is correct? Blood and water flow in a concurrent. No, that's not right. It's countercurrent. So that's not right. Blood and water flow in a countercurrent system with a constant concentration gradient between them. I think this is correct. I'm just going to read on though, just make sure. C. No, not concurrent. No. Countercurrent is yes, but with a greater no. Okay, yes, it is constant. B. And that's because if you remember the diagram, it looks a little bit, I'll just draw it down here, it looks a little bit like this. This is percentage O2, and we have the blood kind of flowing this way, sort of getting more oxygenated, and we have the water flowing this way, getting less oxygenated, if this is distance along gill filament. Okay, so therefore it's B. All right, moving on. 8. Rubisco is an enzyme that carries carbon dioxide in photosynthesis. In some conditions, it can also carry out oxygen fixation. This is called photorespiration. It's bad. The graph below shows how the carbon dioxide and oxygen fixing abilities of Rubisco are affected by temperature. Okay, so first of all, look at the legend. Carbon dioxide fixation is the dotted line. Oxygen fixation is the sort of what's it called? The solid line. What are the correct percentage changes in Rubisco carbon dioxide? Oh God, between 30 and 40. Okay, 30 and 40. There's going to be a bit of mass here. All right, carbon dioxide fixation goes, well actually this is going to be an easier one than you think. Carbon dioxide fixation at 30 is there, and at 40 is pretty much the same, isn't it? So it might go down a fraction. Carbon dioxide fixation is down a tiny fraction, so I'm going to say that we don't even have to do the maths. It's going to be one of these two. One of those two, down a small, tiny amount. Okay, so for the second bit, the oxygen fixation, I think we better do a calculation because it could be easy to get this wrong. So oxygen fixation at 30 is exactly, and if we move along here, it's there. So that is 2.4. And then at 40, it's probably about there. So I would say that is 3.246 is 3.7. So what percentage is that? So 2.4 to 3.7. So that's an increase of 1.3. And to make it a percentage change, it's 1.3 over the starting value, which is 2.4 times 100%. And if we do that calculation, that's going to be close to 50%, isn't it? So let's just do that. 1.3 divided by 2.4 equals times 100. So I'm getting 54%. Yeah, so that is exactly this. So there we go. The answer is C. Okay, zooming out a bit more. Okay, moving on. 9. The hormone HCG can be detected in urine using pregnancy tests. Which of the following properties of the hormone HCG allows it to be detected in urine? It's not really that, I don't think. Okay, well this is a property, okay, because this means, why is this relevant? Well, this means that it can get out at the glomerulus, it is passed in ultrafiltration into the filtrate, and then it gets into urine. This isn't really relevant, and this isn't really relevant, so it's just B. 10. The hormone ecdysone is synthesized in the prothoracic gland found in the upper thorax of some invertebrates and is released into the hemolymph. It's then transported to cells near the surface of the body and causes the loss of the exoskeleton, so the new skeleton can form. Which of the following statements explains how ecdysone is able to act on cells near the surface? Well, this is just basically how does a hormone work. Okay, there's a lot of information, but really a hormone, how does it work? Well, 3 is important because it binds to the cell surface receptors. And 2, I think is also important. I was thinking, could it be only 3? So I don't know, it can't be only 3, but probably 2 and 3. So it's a hormone, which means it travels in the blood, but insects don't have blood, they have hemolymph. So it travels in the blood and binds to receptors. So I think 2 and 3. Yeah, 10. C is the answer, yeah. 11. Adhesion. This is just a definition question. Adhesion is water molecules sticking to something else, not to each other. To each other is cohesion. It's to the walls of something, for example, the xylem. So, yeah, it's going to be A. Just double check. No, that would be cohesion. No, that's not a thing. And that again would be cohesion in the phloem, so the answer is A. 12. The image below shows the structure of nuclide-based guanine. Bird organisms as guano. Didn't know that. Because they contain a high proportion of guanine. Unlike mammals, birds excrete nitrogenous waste as guanine instead of urea. Guanine synthesized from marine. Okay, following statements refer to guanine. Ammonia is more toxic than guanine. Urea is more soluble in water than guanine. Guanine has a high proportion of nitrogen, which the statement correctly explains why birds excrete guanine. I would say 1 and 3. No, that's not an option. Okay, so you don't want to have ammonia, you want to get rid of it. So that is turning it into guanine, so that's relevant. Guanine has a high proportion of is it all of them? I'm going to say, I think it's all of them. 1, 2, and 3. Yeah, that's the only thing. That's my best guess, and it's right. Yeah, it's all of them. Sometimes you just kind of have to give your best guess. And yeah, all those things are relevant. 13. Different sized mammals have different surface area to volume ratios. Okay. Which of the following options is the correct order of surface area to volume ratio for the different mammals, arranged from largest to smallest? Okay, well, you just got to work out the surface area to volume ratios for each of these things, don't you? Okay, so for example, surface area to volume ratio, so you divide by the volume, so 0.48. Oh gosh, it's so much standard form, I hate standard form. Divided by 2 times 10 to the minus 2 is 0.02. So that's like 24. This is a bit easier. 18.26 divided by, 18.26 divided by 2.24. 8.15. It's also going to be in order of size, actually. So maybe that's a shorthand. A moose is a mouse. This is a rat. If you know your genesis, this is a mouse. This is a horse. Got no idea what that is, to be honest. Autolagus. So we would expect the rat to be bigger than the mouse, which I think is true because it's a bigger animal. Sorry, no, it's a bigger animal, so therefore it would have a lower surface area to volume ratio. So let's do that one. Let's do the mouse one. Okay, 1.9 times 10 to the minus 3 is 0.0019. Yeah, that's right. I'm just trying to do standard form in my head and divide it by, oh my gosh, 7.2 times 10 to the minus 5. Okay, that's 0.12340 zeros, 72 equals, that's a 26-ish. This is a lot of work for 1 mark, isn't it? And the last one is 0.32 divided by 0.016, 20. Gosh, so we should hopefully get this right. From largest to smallest. So autologous is first, then it's, oh no, sorry, mouse is biggest, isn't it? Sorry, mouse is biggest. Then it's the autologous, and then it's the rat, and then it's the horse. So it's C. Checking the mark scheme. Yes, I got it right. God, that was quite a lot of work for 1 mark. 14. The commercially grown tobacco plant, Nicotina rustica, has many pests. One such insect pest is Manduca sexta, which causes damage to the stems and leaves of N. rustica. The tiny wasp, Cotesia congrita, lays its eggs inside the body of M. sexta. When the larvae develop, they feed on the body of the host, eventually killing it. Incidentally, this is the inspiration for the Alien movies. This horrible little wasp parasitizing this caterpillar. It's pretty grim for the caterpillar. Okay, so N. rustica produces a compound, a volatile organic compound called volaticin when its leaves are damaged, and it attracts basically the wasp, which is amazing. So it kind of signals the predator to come and kill the caterpillars. That's pretty cool, isn't it? So volaticin release reduces herbivory. No. Volaticin release increases M. sexta growth rate. No. Volaticin release reduces parasitism. No, that's not. Okay, so going back to number 1, I think number 1 is correct. Okay, it does reduce herbivory because it causes the parasite to come and kill the herbivores, so therefore it is D. 14D. Yep, that's right, good. On to the last multiple choice. Mistletoe. That's Christmassy. Mistletoe is a parasitic plant, and it grows on other trees. Survives by removing water and assimilates. Remember, this word assimilates, it comes up every once in a while, so that just basically also means stuff like sucrose and anything in the phloem from the host plant. The mistletoe binds to the stem of the host plant and grows a specialized root-like tissue called a haustorium that attaches to different tissues in the stem. One species of mistletoe, Viscum minimum, contains no chloroplasts. Why does it not need chloroplasts? Well, it's because it's going to be attaching to the phloem, so it's a sieve tube. Xylem would just get water, it would still need chloroplasts. Attaches to meristem cells, well, that wouldn't really do anything. Attaches to Cambrium tissue, that's another word for meristem cells, really. So A is the only sensible answer, and that is in fact true. It gets sugars from the other plant. Okay, so that's the multiple choice segment done, and now let's move on to the longer answer questions. Okay, here we go. Question 16. Many insects, such as moths and bumblebees, are insulated with scales and hair, are known as facultative endotherms, which means they can kind of act like endotherms. They're not real endotherms, but they can kind of be like endotherms. Their metabolism during flight causes the temperature of the flight muscles to increase 20 to 30 degrees C above the external temperature. That's a lot. Using the information provided, explain why many moths and bumblebees are described as endothermic. It's basically just they can maintain their body temperatures above the external temperatures, or they can get heat energy through their own metabolism as well. 16 part 2 is more difficult for moths and bumblebees to maintain their body temperature than for mammals and birds to maintain their body temperature. Well, the first thing is that they are very small. So they're very small, so they have a high surface area to volume ratio, and they lose heat energy rapidly. Maybe also they don't have they also cannot store as much heat energy internally because they are small. Let's look at the mark scheme and see how we did. Okay, so they generate heat from respiration metabolism. This is what I said. Also, the scales and hair help to reduce heat loss. Allow, they generate heat internally. Ignore, they don't generate temperature internally, because temperature and heat are not the same thing. So for the second part, they are smaller and have a large surface area to volume ratio. That's what they wanted us to say. So they have a greater rate of heat loss. You've got to say greater, notice that there, greater rate of heat loss. Mammals and birds have more thicker insulation. I could have talked about this. So fat under skin. I was thinking about that. So mammals have fat under skin and birds have just a thick layer of... They also have fat under the skin as well, both mammals and birds actually. Reference to method of more precise control of body temperature in birds and mammals, for example, thermoregulatory center. So anything you knew about the mechanism of control of endotherms you could have brought into that question. Okay, on to the next bit. When waking, the abdomen of caterpillars expands and contracts slowly. Air is taken into the tiny holes alongside the body. These are called spiracles. Don't need to consult the mark scheme for that one. Fluid is found in the tubes responsible for gaseous exchange. This is called tracheal fluid. And it can be kind of moved into the tracheal system or out, tracheal system or out, to increase the surface area available for gas exchange or not. Okay, so reasons for transport systems. Well, they are metabolically active. They have an insufficient surface area to volume ratio to get their oxygen, get sufficient oxygen in, and also cells in the center of the animal are too far away from the outside to rely on diffusion of oxygen into the center. So those are 3 points that you can say. I'm sure there's some more in the mark scheme, which we're going to have a look at right now. So high metabolic rate, so they therefore need a lot of oxygen. Could have said that as well. Diffusion is not sufficient, that's what we said. Distance is too far. Surface area to volume ratio is too low. And then we actually should just name the things that they need. So named metabolite, needed by cells. So the perfect answer would be something like the cells in the center of these organisms need oxygen. That's this mark here. But these cells are too far away from the external surface to allow diffusion to work. That would be this mark here. These organisms also have a high metabolic rate and therefore need a lot of oxygen. And additionally, the surface area to volume ratio is too small. So just making sure you get all the marks that you can. Even when it's a 3 marker, try and go for 4, just to kind of have a bit of insurance almost. D. A student planned to carry out a dissection of insect and fish gaseous exchange systems. The student planned to complete diagrams of the different tissues. They're advised to observe the following guidelines. Use a sharp pencil. Use ruled label lines. Include a scale bar. So there's 2 further guidelines for the student to follow to ensure they present their diagrams clearly and accurately. Oh, I hate these ones. Clear continuous lines, I think. Clear continuous lines. And I think make sure the diagrams are large, so they cover at least 50% of the area. And I think no shading as well. I think those are 3 things that might be on the mark scheme, but to be honest with you, I'm not very good at these ones. I always forget. I find these questions a bit tiresome. Okay, right. See, yeah, there's so many boring things. Okay, so 50% of the available space. I did remember that one. Continuous lines, no shading. Okay, those are 3 that I remembered. Here are some other things. State magnification, correct proportions. Label lines should not cross over other label lines. So that means if you're like, I don't know, if we're labeling, here's a fish. This is my terrible fish. And you can't label the eye there and the mouth there. Okay, that would be bad because they're crossed over. Not like that. So there they are. Pause that if you want to have another look at those before we move on. Okay. Okay, we're on to question 17. What am I doing for time? Okay. Cirrhosis of the liver can result from long-term liver damage. Alcohol or other toxins can cause this damage. Scientists suggested that cirrhosis can be detected by taking samples of body fluids and testing them for 2 different molecules, C-reactive protein and copeptin. The liver produces these 2 molecules, and increased levels can indicate liver damage due to cirrhosis. Different body fluids from a patient suspected of having cirrhosis were tested for C-reactive protein and copeptin. Okay, so C-reactive protein is super high in blood, whereas copeptin is seemingly very high in feces, well, and urine as well. That's interesting. Okay, different body fluids have different concentrations of the different molecules. Calculate the order of magnitude by which concentration of copeptin in the feces is higher than the concentration of C-reactive protein in the saliva. Oh, maths. Okay, so copeptin in the feces. That's this one, in the feces. Oh, and it's powers as well. Joy of joys. I'd love to test the maths here. This is about 10 to the 9. Okay? 10 to the 9. And the C-reactive protein in the saliva is C-reactive protein in the saliva is 10 to the 1. Okay, well then it's 10 to the 8. So it's 10 to the 9 divided by 10 to the 8 equals, 10 to the 1. 10 to the 9 divided by 10 to the 1. And when you're doing powers in maths, remember you take away. If you're dividing 10 to the 9 by 10 to the 1, you take away. So it's 10 to the 8. I think that's sufficient. I don't think you have to put that in longhand. Yeah, 10 to the 8, or 1 times 10 to the 8, or you could have written 100 million. So yeah, all those are correct. Here we go. Okay, moving on to the next question. Suggest why blood and feces have the highest concentration of C-reactive protein and copeptin. So why does blood have the highest concentration of C-reactive protein, and why does feces have the highest concentration of copeptin? Okay, well, this is interesting. We have to go back and think, where are these made? They're made in the liver. The liver produces these two molecules. So, well, the liver has kind of two routes out of it. It can produce something to be released into the blood, or it can produce something and then it can travel into the bile and into the feces. So I think that basically the copeptin is going to be in the bile, which is going to travel into the small intestine and into the feces. Whereas the C-reactive protein is released into the blood. Back into the sinusoids, let's say, and into the blood. So I think this question is testing us about our knowledge of the liver structure. So let's see if that's right. Yeah, okay, so sinusoids. Yes, I just got that mark. Sort of lucky there. I need to say it has a good blood supply, but sort of talking about the sinusoids and the fact that it's going into the blood, and copeptin is released into the bile. Okay, yeah, so good. B. Kupffer cell from the liver. The diameter of the Kupffer cell in the image is 9.1 centimeters. Assuming it's spherical, calculate the actual volume of this cell. Give your answer to full significance figures, show you're working. Okay, so I'm going to struggle with this one because I can't measure it on my computer screen. But what we're saying, the actual diameter is 9.1 centimeters. So the first thing you need to do is you need to measure this length. You need to measure this length with your ruler and figure out how many centimeters is it. And then you can work out the actual scale. Okay, so from that you work out the scale. So if this was like 2 centimeters, let's say, which it probably is something like that, then it would be 20 millimeters or 20,000 micrometers, and then you can work out the scale. So I'm not sure what the actual value of this is here because I haven't got it in front of me, but let's just follow the working as if I did. So I'm going to say this is 2 centimeters. So then I would say magnification equals the image size divided by the actual. So if let's say the image here, this bar, let's say I'm measuring it as 2 centimeters, that would be 20 millimeters or 20,000 micrometers divided by 4 micrometers. Therefore, that is times 5,000 magnification. And then I could work out what this value is. So 9.1 centimeters divided by 5,000 is 91,000 micrometers divided by 5,000. So 91 divided by 5 essentially. 91 divided by 5 equals 18.2. So I'm saying that that distance would be 18.2 micrometers. But then, this is hard actually. So then, if that's the total diameter, I need to divide it by 2. So let's label this. It makes it much easier for the markers. Diameter equals 18.2. Therefore, radius equals half that. 9.1 micrometers. Then I have to use, this is something you need to know that the volume of a spherical object is 4/3 pi r cubed. Okay, so you plug that in, 9.1 times 9.1 equals times 9.1 equals times 4 equals divided by 3 equals, and then times pi, there's pi equals, and I get 3,157 micrometers cubed. Is that actually right? If I just guessed that was the right? Yeah, I did get it right. Okay, cool. So yeah, 3,157. So therefore, on your thing, this must have been 2 centimeters, which I just guessed. Okay, so that's how you get there. It's quite tricky. Which type of microscope has been used to obtain this image? Okay, well, if you go back and look at the image, that is definitely an electron microscope. Then you have to decide, is it transmission or is it scanning? And it is transmission, because scanning gives you the 3D. So it is transmission, TEM, I'm just going to write for short. You should write transmission electron microscope. And then how do you know? Well, there's a great amount of detail. You can see mitochondria. You can see things that are smaller than 1 micrometer. There's a high resolution, and that should be sufficient. So let's double check the mark scheme for that. So 1 mark for transmission electron microscope. In fact, just electron was okay, but you couldn't, if you put scanning, you would have wrong. Except these, you can see ultrastructures, there's a high resolution and a high magnification, so there's lots of things you could have said there. Okay, let's move on. Let's do question 18. And then maybe we'll do a break. Okay, so question 18. Light intensity, carbon dioxide, and temperature are all limiting factors in photosynthesis. What is a limiting factor? Well, this is a factor that can limit the rate of photosynthesis. That's not a very good answer, is it? It's kind of repeating the thing. It is factors that when there's not enough of that factor, it can slow the rate of photosynthesis, and you can increase the other factors and it'll have no effect. That's what I'm saying, but let's just look at the definition here, because I think that's probably going to be better. Okay, so the word they're looking for is rate. This is what their examiner's looking for. It's underlined. The factor that will determine the rate of photosynthesis. Not amount, but rate. I think I would have got that. When at a lower level. When at a lower or suboptimal level. So yeah, maybe learn this definition. So what is a limiting factor? It is a factor that will limit the rate of photosynthesis when at a suboptimal level, or when in the shortest supplies. You could have got that. Moving on. Okay. So when faced with a question like this, we've got a bit of data about an experiment. First thing is to try and kind of get an idea about the experiment, about what's going on. So the investigation was carried out into the effect of adding different volumes of water onto the survival of seedlings. 60 seedlings in each group. And the results were shown in Table 8. So if the conditions are good, you'll expect all potentially the seedlings to survive. So volume of water added to the soil, and it goes up to day 18. So this is going to be important here. By day 18, 57 of the 60 are surviving with 10 centimeters of water. So that's pretty good. With 20 centimeters of water, by day 18, 53, so fewer. With 30 centimeters of water, only 48 out of the 60 are surviving. With 40 centimeters of water, only 20 of them are surviving. And with 60 centimeters of water, only 2 are surviving. So this, basically you're drowned. As you're adding more water, the seedlings are kind of getting overwatered and are in fact sort of drowning in a sense. So is there any other data that we can sort of pick out? Like sort of how long does it take the seedlings to die? Typically they do all right. Well, I guess it's a relatively steady decrease. I mean, these ones sort of actually seem to survive for longer. So with smaller amounts of water, that's pretty, just a small decline and pretty steady. With 30, they survive quite well up until about day 9, and then they sort of kind of drop off quite rapidly. With 40, they survive, well, they start dying straight away, really, but they do all right, I guess, up to about day 6. And with 60 centimeters of water, well, they just start dying straight away. So, yeah, that's it. Overwatering kills the seedlings, essentially, is the conclusion. But now let's see what the questions are about. Summarize the conclusions. Okay, well, first of all, more seedlings survive with 10 centimeters of water. Okay, most seedlings survive with 10 cm of water. I need 3 marks for this. We could do some percentages, so 57 out of 60 as a percent. We could do that. What percentage is that? It's very high. Let's quickly do it. Because there's 3 marks, so there's going to be probably some marks for kind of working out some numericals. That's 95% actually. And then let's do the next one, which is 53 out of 60. So this is 88%. 48 divided by 60 is 80%. 20 divided by 60. That's a big drop. It's 33%. Okay, so this actually doing percentages is helping because we can sort of see where the major. That's only 3% or 3.3%. So this up to this point seems okay-ish. So it seems to be the effect is more pronounced above 30 centimeters cubed. Most things survive with 10 centimeters of water. Watering more than 30 cm cubed of water has a severe impact or something. And then I'd quote some data. I won't write it here, but I'd say, for example, 30 centimeters cubed of water results in 80% surviving at day 18, but 40 centimeters of water results in only 33% surviving at day 18. So quote some data there. And finally, maybe you could say when the seedlings are overwatered, the death seems to be most significant after day 9, maybe? Oh no, maybe after day 6. I don't know. I mean, that's what I can do. Let's see what the mark scheme says. Okay, here's 3 conclusions. Increased volume of water added leads to lower survival. Okay. Larger decrease. Okay, we said that. Larger decrease in survival for added above. Volume of water has no effect on number of seedlings for surviving up to the first 3 days. Okay, this is one I didn't get. And quote data points. Okay, well, I did quote some data points, so that's okay. We did talk about this. I think I forgot to say the obvious. It's sometimes very easy to do. Didn't actually say the obvious thing, which is that with more water, there is a lower survival rate of seedlings. I just said that the optimum was 10, which I'm not sure if I would get a mark for that. So always say the obvious. It's sometimes so easy just to forget about the obvious thing. Okay, let's go back to the question. I'm just going to finish this question and we'll take a quick break. Water can fill air spaces in the soil surrounding the roots. This prevents oxygen from reaching root hair cells. Use your knowledge of aerobic and anaerobic respiration. Explain why overwatering can kill plants. Now, if you're in my class, I gave you a bit of a clue before you did this because I said that anaerobic respiration is less ATP produced. So let's follow this through. So if we have low oxygen, that means that anaerobic respiration occurs instead of aerobic, which means we have less ATP produced. If we have less ATP produced, we have less ATP for active transport. So if we have less ATP for active transport, we can have less mineral ions into the roots. And paradoxically, therefore, we can have less, not more, water moving or following via osmosis. So therefore we can have sort of less root pressure and sort of less water in the xylem, which is so weird because you think that you give lots of water and it has lots of water, but no, it actually means that it can't intake the water. So that's what I'm thinking. And of course, any other metabolic processes in the roots are also going to suffer. So that's going to be an issue. Let's look at the mark scheme and see what else that I've missed. Okay, so no oxygen, so no aerobic respiration occurs. Further detail. So we haven't, in my class, we haven't covered respiration yet, but we will. So this is some other stuff that you could have said. You could explain why there's a less ATP because, we'll learn about this later, there's no link reaction, no Krebs cycle, and no oxidative phosphorylation. So this is extra stuff that we will cover. In anaerobic respiration, plant has to switch to anaerobic respiration and there's less ATP produced. Less ATP produced, so no active transport, cannot take up mineral ions, so cannot generate water potential gradient. So this is less root pressure and actually weirdly less water can be absorbed. So less water gets into the plant weirdly. Okay, so maybe you could probably get my class could have got sort of 4 or 5 out of 6 there, but I know we haven't covered respiration yet, but we will. So in order to get 6 marks, let's look at the descriptor up here. What we need is a detailed scientific statement about aerobic respiration and a detailed scientific statement about anaerobic respiration. And more than one scientific consequence. Okay, so I went heavy on the consequences because we haven't done the detail yet. So let's say if you just followed the consequences through enough for my class, you can give yourself a couple extra marks. But if you're watching this and you're not in my class, that's what you need to say. You need to talk about the link reaction and Krebs cycle not occurring because we don't have that final electron acceptor. So therefore the whole system backs up. We have no electron transport chain, no ATP synthase working via sort of chemiosmosis and so on and so on. So therefore the whole system kind of breaks down. So pause that there and have a look at all those marks. I'll zoom out just a little bit more so you can kind of get it all on one page. That is quite big. And pause it there and have a look at that mark scheme. Okay. Right, so that was question, one more, and then we'll finish. Okay, soluble mineral ions are present in the soil. Explain why water molecules can form hydrogen bonds with nitrate NO3. Well, they're polar. Polar molecule. Edit. Okay, so here I've dropped in a picture of the structure, and actually this is kind of sort of delocalized. You don't need to know this if you're a biologist, but if you are a chemist, this might help you a little bit. So this is kind of almost like a half bond, and this is a half bond, and this is a half bond. But so the oxygens are negatively charged, which means that water, if we draw water here, you can form a hydrogen bond here. Okay, interaction between the delta plus hydrogen and the delta minus or the minus oxygen. And if we were to have a water molecule sort of above, water molecule here, you imagine this is sort of floating slightly above, it could interact with the nitrogen there. So that is the reason. And on the mark scheme, we say water is a polar molecule, and nitrate is charged or negative, and hydrogen bonds form between the H on water and the O on nitrate. Note the following examples. So you could actually give that structure. Look at them all here. So drawing the diagram actually like I have done would help. Okay. And back to the last bit of question 18. Figure 18 shows a process that occurs in the cell surface membrane of the endodermis in the root. So explain how this causes water to enter the endodermis. So we have active transport of mineral ions. So these are mineral ions. And the diagram gives us a lot of clues, so we best sort of use those clues. So we have active transport moving from a low concentration to a high concentration of mineral ions, which means that we have a low water potential in here because we have a high concentration of solute. So therefore water moves from a low water potential, I'll use blue for water. Water then moves via osmosis from a high water potential, low solute, to a low water potential. So this is osmosis. And that should definitely be sufficient for 2 marks. Double check the mark scheme. Here it is. Solute ions or mineral ions against concentration gradient by active transport. Reduces water potential of the cells. Water moves down water potential gradient. I'm not entirely sure if I would have got this. I think I might have said the root. But you need to talk about the root cells, the endodermis cells. D. Okay, this is really about the definition of an organ. So we have multiple tissues. And then maybe I'd sort of go into tissue is a group of cells with a similar structure that have a certain function. And then I'd probably give examples. For example, palisade mesophyll. And maybe give one other example. For example, upper epidermis maybe, or the spongy mesophyll, pick one. And then say that an organ is when we have multiple tissues all working together to do one job. And then we say that the job of the leaf is, of course, photosynthesis. So let's check the answer. So organ is a collection of tissues, yes, performed to carry out one role or one job. And the leaves have 2 tissues. We can pick some of these tissues and explain. So epidermis, spongy mesophyll, and so on and so on. Okay, pause that there if you'd like to have a bit more of a look. Okay, so we are about 40 minutes into this video, so this is a great place to have a break before we move on to question 19. So I recommend go grab a cup of tea, take a little break, come back, pause this, and then we will resume with charging through this A-level paper. Okay, see you in a sec. Okay, so question 19. Sperm cells are formed by a process known as spermatogenesis. So this is a meiosis style process. So here we have the summary. So we can see the sort of, let's just sort of annotate this diagram. So we've got the, copying of the DNA is here. And then at B, we've got meiosis 1. Because we can see we're going from individual kind of chromosomes to the double chromosome. So this is copying the DNA. This is meiosis 1, the first division, where we separate, this one and this one. Those are the homologous chromosomes, and they separate to here and here. And then this is meiosis 2. And I don't know what this is. This is just putting a tail on it. And then making it into a sperm cell. Okay, so metaphase 1 occurs during the stage labeled B. Telophase 2, that's going to be in meiosis 2, so that's labeled C. And anaphase 1 occurs also during B. Okay, double check the mark scheme there. BCB, yeah, great. Okay, the chromosomes carried by sperm are made of DNA. Nucleic acids are made from what monomers? Nucleotide. Phosphodester bonds formed between the monomers. They consist of a phosphate group between the, we're talking about between the pentose molecules, because this is generic nucleic acid. So we could be talking about RNA or DNA, so hence not deoxyribose or ribose, just pentose. In DNA, hydrogen bonding between the two antiparallel strands, yep, there it is, strands, causes the characteristic double helix shape. Easy marks there. Moving on. C. Figure 19.2 is a transverse section of a sperm cell. So what this means is, if you can't visualize it, here's the sort of head. This is the tail. Oops, there's a bad tail. But the transverse section means it's been sort of sliced here, and we're looking at the kind of, if you like, imagine if you cut a sausage in half and then sort of look at it end on. So what are the structures? This is interesting. This does come up in your syllabus, but I reckon that some of you might not be aware. U is the mitochondria, actually. So the mitochondria actually wraps all the way around this central arrangement of kind of microtubules here. Actually, you're not required to identify that. What's W? Oh, so this is, okay. They're all mitochondria, but what part of the mitochondria? So U is the mitochondrial matrix. W is the mitochondria, the crista is one of them, isn't it, I guess. And finally, Z, intermembrane space. Okay, I realise that this is on respiration now, so for my class, sorry about that, I didn't spot this one on respiration. But we're going to go over this later. Okay, the next part, let's double check that mark scheme first of all. Matrix, crista, intermembrane space, yeah, great. Right, ATP, FADH2, and hexose 1,6-phosphate are three organic products of respiration in sperm cells. Table 19 shows how the production of ATP, FADH2, and hexose 1,6-phosphate in sperm cells is affected by three different substances. Organic products of respiration. Okay, so cyanide, fluoride, and sucrose. Now this is annoying because it doesn't give you a kind of control. Well, I guess sucrose is the control. So sucrose is just the sugar. So under normal conditions, sucrose, remember, is a disaccharide, and it contains glucose plus fructose, but it can be easily hydrolyzed into glucose. So this is basically just sugar. So we have 6.89, 2.53, and 5.42. So then with the cyanide, we can see that there is still some ATP produced, but there's zero FADH2, and there seems to be a sort of normal amount of this. This is drastically reduced, and fluoride just knocks out everything. What can be concluded that difference between effects of cyanide and fluoride on respiration in sperm? Okay, so this is pretty simple. So once we've done the subject, that is, for my class, so it is that cyanide stops or blocks aerobic respiration only, whereas fluoride minus blocks aerobic plus anaerobic respiration, stops respiration entirely. Mark scheme check. Yeah. Cool. Moving on to question 20. Okay, so this question here, question 20, very sort of data intensive. So the first thing we should do is kind of get a feel for the table to try and figure out what's going on. So if we look at the table, we've got 5 different types of yeast. Here they are: S. cerevisiae, C. krusei, C. albidus, C. albicans, and A. pullulans. And we're looking at their rate of CO2 production and we're looking at it in aerobic conditions, which is with oxygen, and we're looking also at anaerobic conditions, which is no oxygen, with no oxygen. So now annoyingly we don't have the means on the table, but we do have the standard deviations. So where are the means? It does say above the table here. I hope you saw it when you read it. Table 20 shows the results of the experiment. The mean values for these data are represented as a graph in figure 20. Now the first question here is to calculate the standard deviation. So therefore, you can do this straight in your calculator if you know how to use your graphic calculator, or you can work it out sort of longhand on the page, and that's what I'm going to be doing. But for the standard deviation, first of all, we need to remember the formula. So standard deviation, SD, equals, so it's the value x minus the mean, which we call x bar, and then we square that, and then we sum all of those, and then we divide it by n minus 1, and finally, I haven't really left enough space, we have to do a square root of the whole thing. So let's do that on the table. So the standard deviation that we're looking for is this value here. So first thing we need is the mean, because we need to do, let's just do this, minus the mean. So I'll make this little column here minus the mean, and then we will square it, 2 there for squared, and then we're going to total them up, and then yeah, we'll work it out. So first thing is we need the mean. So the mean for this Pullaland's anaerobic conditions we can find is the anaerobic conditions is the sort of shaded one, and Pullaland's is here. So the mean there is 30 is the mean. So now let's get cracking, and I'm going to edit this to make it a bit faster. So 34 minus 30 is 4, 6, 1, 2, 2, 4, 3, 1, 5, 10, 10, and 4. Now I haven't put the signs on that because of course when you square it, whether it's negative or positive is irrelevant. So it's not necessary really. So when we square it, we've got, oh, this is a bit of mental maths for me here, we've got 16, we've got 36, 1, 4, 4, 16, 9, 1, 25, 100, 116, and then we're going to total those up. So that I'm definitely going to need my calculator. So I get my total is 328. So I've got 328 over n minus 1. Now what's n? So there's 12 trials. So n minus 1 is 12 minus 1 equals 11. So it's 328 divided by 11 equals square root of about 29.8. And I'll do that. So I'm going to get, where is that, square root, 5.46. All right. So there is my answer, but it says, always check this, give your answer to one significant figure and put it on the table as well. So let's do that. So the examiner is going to mark this answer right here. And to one significant figure, the answer is 5, okay? 5. There we go, let's double check the mark scheme. Now the mark scheme here, 3 marks. And actually they do allow 3, 4, or 5, or 6, strangely, if the working is correct, because you may have used this formula n minus 1, which is the formula which I used, divided by n minus 1, or I think in maths, sometimes you use divided by n, so I think they give you leeway depending on which formula you used. So, there we go. Now the next thing is to plot the standard deviations for all the data on figure 2. So remember when we're plotting the standard deviations, we use error bars plus or minus the standard deviation. So I'll just do one of these to show you how, and you would need to do them all like this. So for example, S. cerevisiae aerobic, the standard deviation here is 4. Right, that was S. cerevisiae aerobic. So over here, S. cerevisiae under aerobic conditions, we start at 21, so we would mark the standard deviation +4, and we would also mark the mean +1 standard deviation and the mean -1 standard deviation as well. So that's 2 and that's 4, so that's there. So that is how we should do it. Oops, I haven't drawn that brilliantly, but we draw a sort of error bar like that. And all of them should be like this. So they should sort of go up and down like this. These are not accurate. I'm just sort of showing you what it's going to look like. I haven't actually checked the figures. But let's look at the mark scheme now and see what exactly you need to do to get those 2 marks. It just says SD bars plotted correctly for the first 4 yeast species above and below the mean. Okay, moving on. 20C. Calculate the mean percentage change in CO2 production for S. cerevisiae when moving from anaerobic to aerobic respiration. Give your answer to 4 significant figures. So we've got S. cerevisiae, so we need to go up to the table, and we're moving from anaerobic to aerobic conditions. So first of all, we need to extract some data from the table. So here, this under anaerobic conditions, S. cerevisiae is at 13, and under aerobic conditions, it is at 21. So we're going from 13 to 21. So from 13 to 21. Why have I changed colour there? I don't know. So that is a change of 8. So it's +8. And remember percentage change, you've got to know this formula equals the change divided by the starting value times 100 to make it a percent. So therefore it is 8 divided by the starting value of 13 times 100. I can't believe this is worth 3 marks. This seems too easy. So 8 divided by 13 equals times 100 equals. And that's always how I put it in my calculator to save me going wrong. 8 divided by 13, press the equals button, times 100, press the equals button. So I've got 61 and 4 significant figures. Okay, so 61.54%, because I've got 61.538 on my calculator, so that is to 4SF. Double check that I've got that right. Yep, 61.54 there is correct. Okay, now let's look at the conclusions of this experiment. The student drew the following conclusions. All the yeast I investigated produced more CO2 during aerobic respiration than anaerobic respiration. We'll check that in a second. 2, there is a significant difference between the CO2 production in aerobic and anaerobic conditions in C. albidus. So first of all the yeast. Well, this rings alarm bells. I bet you there's one which is not the case. Yeah, there it is. There's one which is not the case. A. pullulans, here, it produced more CO2 under anaerobic conditions, so that is clearly not correct. So first thing to say, sort of a no, A. pullulans produced more CO2 in anaerobic conditions and therefore conclusion 1 is invalid or incorrect. First mark achieved. Then this one, significant difference between the CO2 production in aerobic and anaerobic conditions in C. albidus. So, well, no, it doesn't really look that significant at all, really. We're talking about here. So we've only gone from, what, 27 to 25? So that's not very good. So there's a lot of things here. So, again, I'd say no. There's been a change from 27 to 25. So that's a very small percentage change. We could calculate the percentage change, which would be 2, the change, divided by 27. Let's just do that real quick. 2 divided by 27 equals times 100. So that's only a percentage change of about 7.4%. And we don't know if it's significant. This is the key vocabulary. We can't tell if it's significant without doing a statistical test. So again, that conclusion is not correct. Let's check the mark scheme. So incorrect because, yeah, producing more CO2. I think I would have got this mark, I should have noted, I hadn't plotted the error bars, so that's, I missed that obvious thing. But here I said allow no t-test carried out. Maybe I wouldn't have got that mark, so I just said a statistical test, but significance is to do with statistical tests, or of course the error bars. So what it would look like when you've plotted your error bars, let's add that on there, is, well, I've kind of sort of estimated it here. So because these, let me just draw them properly. So what is the error bars for C albidus? We've got a 7 and a 5. Okay, so that's quite severe. So 7 is going to be 1, 2, 3, 4, 5, 6, 7. That's way up there. And 1, 2, and then down to here. So the error bar is massive there, all the way up and down like that. And then this error bar is 5, so it's up to plus 30 and down to 20. So we really can't say there's a significant difference there because the size of those error bars are massive and they overlap hugely. Okay. The student found the following definitions of errors in a textbook. Still on this question. So random errors, mistakes during measurements caused by low resolution equipment. Systematic errors, repeated inaccurate measurements in the same direction caused by problems with equipment. Which type of error is suggested by the student's data? Justify your answer. That's a tricky one. Let's go back and look at the data. I'm not quite sure what we're getting at here. I've got to say I'm a bit stumped on this one. I'm just going to go look at the mark scheme because I've got no idea what it's getting at. Got baby brain. Random error because some experiments. It's about the SD bars. Because there are large standard deviation bars, it means there's random error. Right, yeah, of course. So let's just kind of illustrate that. So what would it look like? So I'm going to just draw down here, in fact. So here's our kind of table, and I'll just draw 2 bars like this, okay? So with large random error, we have big standard deviations, like this. However, if we were to have a large systematic error, these 2 bars would just be out. So let's say if the systematic error made the bars look shorter, let's say, then the bars would they'd be kind of like that. So this would be a systematic error. And potentially on the system with a systematic error, we could have quite low error bars, but just each kind of average figure would be dropped by a sort of a systematic amount. Whereas with large error bars like this, it implies random error. That is an interesting question. I haven't seen a question like that before. That's a nice one. Finally, we can see 20E here, just the answer given to us in the mark scheme is ribosomes. Yeah, synthesizing these enzymes. Remember, enzymes are proteins. So it's the ribosomes where all protein synthesis occurs. Okay, moving on to question 21. Plant hormones affect the growth of plant tissue in different ways. One such effect is to promote the formation of seedless fruit. Cytokinins are a group of plant hormones. The commercial plant hormone firm carried out research into 3 different cytokinins. So there are different types of them: kinetin, zeatin, and dietin. The firm investigated the effect of adding different volumes of each cytokinin on the production of seedless fruit. The cytokines were sprayed on the flowers of different plants. Over time, the mass of seedless fruits produced by the plants was measured. Okay, oh, this is a bit weird. The axis is strange. It's very strange. I would have expected the volume of cytokinin would have been on the x-axis and the mass of fruit would have been on the y-axis, but this is a completely bizarre way of showing this. So you kind of have to think about it moving along this axis this way. So as the volume of cytokinin applied here, the mass of seedless fruit first kind of doesn't change much, then it changes rapidly, and then it starts to plateau. Here it changes quite rapidly straight away from low concentration, then it starts to level off. And this one seems to just keep on having an effect as we move up this way. Strange graphs there. On the basis of these results, the firm decided to use Dietin in their new plant spray. The firm made the following claim on their packaging. Dietin is scientifically proven to cause production of seedless fruit when applied to flowers. Ugh. So, okay, well, the conclusion is going to have some things for it and probably quite a lot of things against it. So first of all, let's think about the positives. So evaluate means plus, minus, and conclusion. Okay, so that's what we want. I reckon we might be able to say two things for. Maybe we might be able to say 3 or 4 things against, and then we're going to want to make a conclusion as well. So that's the kind of structure that I'm going for. I haven't looked at the mark scheme yet, so this is just my strategy for answering this. So first of all, let's look at the plus on Dietin. So that's C. So yeah, okay, for the first thing we say is that as the volume of cytokinin applied increases, the mass of seedless fruit harvested increases. There is a clear sort of association between those two. As we add more cytokinin going up the y-axis, this mass of seedless fruit harvested also increases. So there is a correlation or clear link. So as volume of Dietin increased, so does mass of seedless fruit. Right, now there's so many negatives here. I guess maybe one more positive thing you could say, just in case, seems to be a directly proportional relationship, roughly. Don't know if this is worth marks, but you might as well say it. I.e., it's a straight line relationship going through 0, 0. All right, negatives, so many. Okay, first of all, correlation is not causation. It's worth saying, because you don't get marked negatively for this. So worth saying, correlation, not causation, because they said cause here. There may be other factors involved, maybe other causal factors. Third of all, we don't know how many fruits they've tested it on. And they've, does it work on all fruits? On all fruits or all flowers? Another thing, we don't know the concentration. They just added different volumes of the cytokinin, but we don't know the concentration. Don't know what concentrations it works at. And equally, I don't think we know the timeline, do we? Yeah, we don't know how the timeline of fruit production was measured as well, so we don't really know much about their methods. So maybe that's worthy of something, timeline kind of mark. And in conclusion, basically, this statement is not robust. It is not valid. Invalid statement. Just sort of articulate that, explain why. Okay, so I've sort of tried to go for 2 positives, 4 negatives, and a conclusion. Let's have a look at the mark scheme and see how I would have done in this answer. So let's first of all look at the level descriptors. I'll zoom in a little bit here. There's loads of things I missed. I can already see things I missed. So for level 5 and 6, you can do a statement in support of the claim and a statement against the claim and more than one comment on the validity of the claim. or actually, this is what I did here. Sorry, this one. I did say one statement in support of the claim and more than one statement against the claim, and then I commented on the validity. So I sort of went for 2 pros, 4 negatives, and a validity statement. So I think I would have got this. All right. So I did say this: as the volume of dieting increases, the mass of seedless fruit increases. That's the only real thing that they've said is for. Against, there's loads. Okay, well, I did mention this before, but I didn't put it in my answer. The labels of the graph are the wrong way around. It's really weird. Correlation is not causation. And so I did say 2 of those. Yeah, I missed loads of these. Look, I should have put this. There's no scale, no error bars. Should be percentage increase instead of just mass. All those things I missed out. Very silly. Bias, yeah, because it's a commercial company trying to sell its own stuff. I did talk about this. No method, no species, concentration not specified. So much stuff is not specified. Look at all this stuff that hasn't been specified. Wow, no control group, no repeats. Oh my god, this is an awful, awful experimental claim, isn't it? With no real data. So definitely it's an invalid claim. I think I probably would have got 6 marks for my sort of strategy, but I'll just flick through that top bit of the mark scheme there, so you can quickly pause if you want to read that top bit of the page. And then furthermore down below, look at all these things to do with the validity of the experiment that they've got. Okay, so again, pause there and have a look at those. Did you mention any of those things? Okay, so yeah, all in all, a very, very poor scientific claim. All right, let's move on. I think okay, so B. Another response affected by plant hormones is phototropism, which is the bending or growing towards light. Now, it's going to give us a method here, and often when you get a question like this, when you've got these bullet points of a method, it's asking you, like, what's wrong with the method? So we kind of have a sort of eye on that as we're reading through it and maybe highlight things we don't like. So 50 cress seedlings, or just highlight things that are important. 50 cress seedlings put on a sterile paper towel, 10 centimeters of water, distilled water, okay. Repeat for 3 different sets of seeds. Set 1 is placed in a box to prevent light shining on the seeds, so fully dark. Set 2 is placed in a box with light from above only. Set 3 is placed in a box with light from the right-hand side only. So, yeah, I mean, sort of maybe what angle is it? Is it 90 degrees to the right? Other thing, we've got light from above, light from the side. It doesn't actually mention light, sort of what type of light? Sort of light control? Is it the same amount of light that's being shone on these 2 boxes? 25C, that seems all right. After 72 hours, remove 20 of the seedlings and count. That's interesting. Okay, so 50 seeds grown, 20 counted. Well, how do you know which ones to count? For example, to illustrate, this is kind of what this experiment might look like. So, except this is, light from the left. So we have light from the left. Maybe no light was this one. And then we have light above, maybe here. Now, if we were measuring 20 of these seedlings here, how would we pick them? Would we pick, let's say, this one, which seems to be growing fully to the left, or would we pick, I don't know, this one? Maybe if we only selected these ones over here, we'd have a lot of bias incorporated if we're not selecting these ones over here. So those are some things we might be thinking about. So first of all, limitation. Maybe how to select 20. Explanation, we just elaborate on the idea of bias, and you might be picking the most bent ones. An improvement, we could use some sort of random selector. Somehow, I don't know, you'd have to number all the seedlings and then use a random number generator, I guess. That's how you could do it. Limitation 2, sort of no control, make sure the light is the same. And let's make that more scientific. Make sure the light intensity and or color or wavelength of light is the same. So, the amount of light would affect the phototropic response is the explanation. And the improvement is, you maybe use a light meter to ensure that the light coming from the right is the same intensity as the light coming from above, or something like that. Other ones, of course, another one which probably you could have said is no measure of the angle of bend. So I'd use a protractor. So protractor is something a little bit like this. It's got a little kind of X marks the spot there, doesn't it? And maybe if you just line it up, make sure your crest seedling's kind of behind it, and then you could kind of use that to measure, I don't know, the angle, which maybe would be about 30 degrees of that seedling. So there's lots of things that could be improved on in this method, and I'm sure there's going to be more in the mark scheme if we have a quick look at that. So here's the mark scheme. Oops, it's jumped all the way to the top for some reason. Where's this crest seedling thing? Here, 21. Okay, so stuff to do with light. Light intensity is not controlled. Size of whole of box, there's a simpler way of controlling it. So different light intensities could lead to changes in phototropism. So we need to control it. So maybe a light meter or bulb of the same wattage, for example, same distance from lamp, same wavelength of color or color of bulb. Those are all things to do with light. Selections of the crest seedlings. So this is sort of unfair because it could lead to bias, and we would need some sort of random selection to select 20 out of 50, because we could, if we weren't careful, if we weren't using a random number generator, we'd only select the 20 most bent, which would introduce a lot of bias. And then, we also talked about a little bit at the end about degree of bending. So maybe measure an angle of bend by using a protractor, for example, because just counting the number of bent ones is not fair. There's other things here which would we miss? Oh, of course, replication. Each experiment is only done once, so that's not really valid. We should do it 3 or 4 or 5 times maybe. It says at least twice here. Size of Petri dish, maybe that has an effect. Could do. Didn't specify that it was the same size as Petri dish. Potentially maybe one dish was small and the seedlings were densely clustered together, which might have an effect, and another Petri dish could be large and they were more spread out, and that might introduce a variable that was not controlled. Okay, let's move on. I think this is the last question, so here we go. Right, on the insert, which I'm going to show you in a second, which letter identifies with the region with the highest hydrostatic pressure, and which 2 letters identify regions that do not contain plasma proteins. Okay, so even before I'm looking at the insert, I'm thinking about this. This is in the kidney. So high hydrostatic pressure, I'm going to be talking about the glomerulus, does not contain plasma proteins. So this is basically the PCT, or once the filtrate has exited the glomerulus, because the plasma proteins are, remember, retained inside the glomerulus because they are too large to get through the basement membrane. So let's have a look at the insert, shall we? Here it is. So the glomerulus is clearly A or D. Oh, is it A or D though? Okay, well it's A, because A is the capillaries in the glomerulus, whereas D looks to be the sort of Bowman's capsule, where the sort of filtrate has been forced into. Now C, I think, is something like an afferent or efferent arteriole. Don't know which one, because there's loads of blood cells in there. So this is some sort of arteriole. So that's definitely got plasma proteins in. So this A is the answer for high hydrostatic pressure. Hydrostatic pressure is A. Now, where there are not plasma proteins, it could be in B. I think it might also be D though. So I think you might be allowed B or D. Now, I'm not entirely sure what B is. It looks like the lumen of the proximal convoluted tubule to me, but I would probably go with D. I may be wrong, but I'd probably go with D. Let's look at the mark scheme for that. So, okay, yeah, either one would have been acceptable. So A for the first one, and B or D for the second one. Okay, 22 part B. Studies of the cell surface membrane of the distal convoluted tubule have provided the following evidence. Okay, sodium potassium pumps. They move potassium ions from the blood to the tubular fluid. They move sodium ions from the tubular fluid to the blood. Use ATP in these processes. Sodium calcium co-transport proteins move calcium ions from the tubular fluid into the blood, move sodium ions into the tubular fluid. Use electrical sodium oxidase process. Oh, this is the distal. Okay, I was about to say this seems very unfamiliar to me, but that's because it's not on the syllabus, the distal. So this is a classic thing when they ask you to compare what you know with something that you don't know yet, but they've just told you. So I'm just going to quickly draw what we do know. Okay, so PCT. So for the PCT, we've got these sort of cells that line the PCT like this, and this is the blood, and this is the lumen. Okay, so in the PCT, what we have is we have co-transport proteins that kind of bring in sodium together with glucose or amino acids, and then that kind of flows that way via diffusion. But what's happening, what's driving the process, sorry, I probably should have drawn that first. The thing that's driving the process is that sodium ions are pushed out and potassium ions are pushed in via the action of the sodium potassium pump. Now, let's compare that to what it's just talked about. So this is prox. Now let's talk about the distal. So what they just told us, in the distal convoluted tubule, I'll just sort of draw the cells here, is that sodium potassium ions move potassium ions from the blood to the tubular fluid. So this is the opposite direction. So K plus is going this way, and sodium is going this way. So that's opposite way round. And ATP is used in the process. Okay, so that's the same. And then move calcium ions from the tubular fluid to the blood. And then move sodium ions back into the tubular fluid. Okay, so now this is like, okay, so what's going on then here is that essentially we've got another co-transport protein, but it's using the gradient of the sodium, but it's coming back out, but at the same time the calcium's going in. Okay, so comparisons. ATP is used for NAK pump in both circumstances. Okay, so that is a similarity. However, NAK pump direction is reversed. So in the proximal convoluted tubule, sodium is pumped out into the PCT lumen, but in the distal convoluted tubule, sodium is pumped into the blood, which is the opposite direction. And then another similarity, I suppose, is that sodium ion gradients are used. So in the proximal convoluted tubule, the sodium ion gradient is used to reabsorb glucose and amino acid, but in the distal convoluted tubule, the sodium ion gradient is used to reabsorb calcium. So that's a difference. So these are the similarities up here. So there's differences down here. So we've got sort of symport. I'm not sure if these words are on your syllabus, but we've got symport versus antiport. I'll explain what those mean. So in this case, the sodium and the glucose are moving in the same direction. We call that a co-transport symport protein. Whereas in this one, the sodium and the calcium are moving in opposite directions. So we call that antiport. As the sodiums go out, the calciums come in. So you could say that the ions are moving in opposite directions. So those are lots of things you could have said. And let's double check the mark scheme here. That's a tough question there. Very technical. So both use active transport, both involve co-transport, both involve reabsorption, both involve sodium ions. Differences: calcium versus glucose and amino acids. Co-transport involves ions only. PCT involves ions and named molecules, so glucose and amino acids. Now you have to say ions. Notice you have to say calcium ions or Ca2+, not just calcium. And I don't think they're marked for the sort of simple antiport thing is not there because it's not really on your syllabus, but it is a difference. Okay, moving on. We're almost there. Okay, so nephrogenic diabetes insipidus is a disease of the kidney that affects the regulation of water potential in the blood. One cause is lithium poisoning. Lithium ions enter the kidney tubules through sodium channels. This prevents the cells of the collecting duct from responding to ADH in the blood. Interesting. So if they can't respond to ADH in the blood, you would expect large quantities of watery urine. So large quantities of urine, pale in colour, I think would be the symptoms. It says not give two symptoms. It actually says state and explain one symptom. Okay, so if lithium inhibits the response to ADH, you could say there's fewer aquaporins in the membranes of the cells in the collecting duct, so less water reabsorbed, so therefore more urine produced, which would be pale in colour. So that's the full explanation. Let's check the mark scheme for that. High volume of urine, fewer aquaporins in the membrane, so that's the full explanation. This says ignore reference to the dilute colour, but that would also be the case. Okay, here's a podocyte, nice picture there. Many gaps between the microscopic process form fenestrations in the Bowman's capsule. Okay, so why are podocytes usually unable to undergo mitosis? Now, I don't know if this is in the syllabus, and I'm not sure if I've ever considered it before, but the picture gives a clue. So when a cell undergoes mitosis, you have a cell and it splits into two. Now, this podocyte, how would that split into two whilst maintaining that fine branching structure? It would be very difficult for it to do, and it would damage or remove some of the fenestrations in the Bowman's capsule. So that's kind of my answer, that's my guess. Never really thought about this before, so let's just look at the mark scheme and see if that's kind of along the right lines. Yeah, so this is kind of what I said, 5 and 6. So if mitosis occurred, it would alter the number, size of the gaps of the fenestrations, so it would alter ultrafiltration. But I think I should have really made sure that there was a link to the cell cycle, year 12 content. Always get the content in when you can. So they are already differentiated. They could be in G0. I think that was a key thing that I could have said. They're in G0, resting phase, so they're not undergoing mitosis. So this idea that the shape is too irregular, asymmetrical, I kind of hinted at that. So yeah, lots of good things there. And they gave you that photo to kind of jog your memory or to kind of lead you in the right direction. Final part. Studies show that after damage by infection or injury, it is possible for nephron tissue to be regenerated. Adult stem cells are involved in this process. So what features of an adult stem cells makes them suitable for regeneration of tissues in the kidney? So adult stem cells, the words you've got to use is multipotent. That means that they have the ability to differentiate into several different cell types. Okay, so that's kind of the next part. Differentiate into many cell types. And you might want to just fully explain, just to make sure you're getting the marks, that they can't differentiate it into every cell type, but the kidney stem cells would be able to differentiate into multiple different cell types within the kidney to regenerate the kidney tissues. So that is probably what the mark scheme says. Let's have a look. So adult stem cells are multipotent, do not allow totipotent or pluripotent, because those are embryonic ones. So they can differentiate and become any cell type within the kidney or the nephron. Great. Okay, so that is the end of that. We've got through that first A-level paper that we've gone through. So well done for getting this far. I think we managed to keep it relatively brief-ish. So I would encourage you to go back through the paper, look over it all, reflect on the areas that you've done well in and the areas you've done poorly in, and make a note of those if you're in my class on the Key Stage 5 assessment tracker to kind of guide your subsequent revision. All right, Merry Christmas, and I'll see you next time. Bye-bye.